Although normally associated with decay, microorganisms are also used extensively in the production of food. A bacterium called lactobacillus is used to make yogurt, and yeast is used to make beer. When yeast respire anaerobically, that is without oxygen, they produce ethanol, which is drinking alcohol. Beer is made from barley, which contains starch. Yeast cannot respire starch, so the seeds are allowed to begin to germinate, which causes the starch to break down into maltose, which the yeast can respire. Ethanol is a byproduct of yeast respiration, which creates the beer. After this, hops can be added to flavour and preserve the beer. It is possible to demonstrate the production of carbon dioxide in anaerobic respiration using the following setup. Firstly, it is important that all oxygen is excluded. Oxygen is removed from water by boiling it, and then a layer of liquid paraffin is added to prevent oxygen re-entering. Yeast and sugar are then added, and a delivery tube is used to connect the test tube to a second containing lime water, which will turn cloudy if carbon dioxide is present. A control experiment can also be done in which the yeast is first boiled, which will kill it. The bacterium lactobacillus can be used to produce yogurt. This happens because the bacteria convert the lactose present in milk into lactic acid. Firstly, the milk is pasteurized to kill any microorganisms. Then it is homogenized, which involves passing it through a fine sieve. This prevents the milk separating out. Before a starter culture containing lactobacillus is added, the milk must be cooled to 43 degrees to prevent the bacteria being killed. Once the yogurt has been produced, it is kept in a fridge at 5 degrees to limit the growth of microorganisms that would make it go off. A fermenter is any container used to grow microorganisms. An industrial fermenter tries to do this as efficiently as possible. Industrial fermenters use computers to regulate their temperature and pH and to provide optimum conditions, as well as paddles to ensure the contents is mixed. Since they contain optimum conditions for the growth of microorganisms, aseptic techniques, such as cleaning between batches, are vital to maximizing yield and ensuring only the desired microorganisms thrive. Industrial fermenters can be used in the production of penicillin, which is an antibiotic that kills bacteria.